Okay. Hi, everybody. All right. Chapter 6. Okay. Another kind of long chapter. So let's try to get through it. Oops. Okay. Self-communication. We'll cover chapter 6.1 and 6.2 in this recording, but we'll go through all the sections. Okay. Okay. First things first. Okay. Because we are a trilling cell organism, all our cells have to communicate with all our other cells some way. And we even really have to communicate with the bacteria inside our guts and stuff like that. So we have to communicate. So cell communication really big part of physiology. Okay, there are two ways that cells communicate. Okay, electrical signals, which will cause changes in the membrane potential of the cell, especially the cell membrane. Okay, and chemical signals. Okay, chemical signals are secreted by cells into the intracellular fluid, and it is responsible for most of the communication inside the body. You guys probably understand electrical signals better right now because you guys understand like muscles and nerves and electricity and stuff like that. But the chemical signals is actually more common and more detailed and more varied. So it's actually harder to explain. Okay. Okay. So for every signal, there has to be a receptor. Okay. There has to be a way to receive the signal. Okay. Or if you don't have a way to receive the signal might as well not be there. Right? Like if you don't have a radio, you can't listen to the radio station. Okay. So the target cells respond to the signals, and then the protein binding of the chemical signals requires a specificity, affinity, so it has to be attracted to it. Okay, there will be competition and there will be saturation. And we'll get into more of that in a little bit. Okay, the easiest form of communication for the cell is local communication. Okay, let's take a look at local communication here. Okay, number one, gap junctions. Gap junctions allow for cytoplasm to flow between cell A and cell B. Okay, so whatever signal it is, if it influences the cell, it will influence the cell eventually, usually within milliseconds. Okay, or possibly less. Microseconds probably. So cell communication this way. The other thing, like for example, calcium flow will be an example of this. The other thing, contact dependent signals, right here, okay, require interaction membrane molecules on these cells. So this one has this thing, that one has that thing, and these line and same signals. One of the most famous ones are just contact inhibition signals. If one cell rubs up against another cell, those two cells, when they're touched, they'll stop growing. Like the signal will be like, don't grow to this side because I'm already here. So it will stop growing. And that way you'll get a single sheet of cells, but they won't overlap each other and they won't compete with each other. Okay. Cancer is very famous for wiping out these signals. So now you have cells that grow on top of one another and kill each other, and the cancer cells win that fight. Okay. All right. The autocrine signals, actually, change this a little bit. Because I realize I want you to see me as well. Okay. Okay. Autocrine signaling. Oops. Oh, I thought I paused the recording. Apparently not. Okay, sorry about that. But anyways, autocrine signaling. Autocrine signaling. Okay. Auto self. Okay. Okay. So this cell under whatever condition can send a signal not only to itself but the neighboring cells around it. Okay. And that neighboring cells around it will respond to the same signal that's being sent out. It's an endocrine solution, but it is for very localized cells. So autocrine is a self-signaling, paracrine is signaling around the cells. Okay, very, very close to the cells. Okay. All right. Okay. So the contact-dependent signals are called CAMs. So cellular adhesion molecules, okay, CAMs. Okay. So for longer distance, you need either hormones or neurotransmitters, neuromodulators, etc. Okay. Okay. Long distance signaling is here. Okay. Okay. So long distance signaling, this is the endocrine system. Okay. Endocrine system, and then nervous system, the neurotransmitters, neural hormones. Okay. You'll see this more in the brain. Neural hormones and neuromodulators are the same pathway. Okay. Cytokines. Okay. Cytokines can act as both a local and a long distance signal. So cytokines are peptides, okay, but they're really cellular communication signals of some sort. And there are different families, okay? Interferons, you guys probably learned in microbiology. Interleukins, you guys probably learned that as in microbiology as well. So between white blood cells, okay, colony stimulating factors, growth factors, tumor necrosis factors, etc. Okay. 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 In cell development and differentiation, cytokines are often used as paracrine and autocrine signals. Um, Honestly, that part was the hardest part of developmental biology, learning all these different cytokines and all the signals they did. But you guys don't have to know that. You guys aren't, going that deep into developmental biology. So don't worry about that. Okay, so they differ from classical hormones in that any nucleate cells can produce them instead of just specialized epithelium. Okay, these signals are made on demand. Okay, and the pathways they signal differ. So let's talk about what signal is. All right, so receptor proteins. Okay, a cell only responds to a particular signal only if the target has a receptor. Okay, so if I delete the receptor, I no longer get signal. Okay, that is one of the things that goes on in diabetes type 2. You lose the receptors, you lose the signal. So it doesn't matter how much insulin is in the bloodstream, if you lose the ability to respond to the receptor. If the receptor goes down, then the signaling goes down. Okay. All right. So signaling pathway. The chemical signal, the first messenger is the ligand that binds to the receptor. The receptor will activate the ligand binding to the receptor will activate the receptor, right? And then the receptor will activate downstream pathways, okay? And then the last signal molecule will modify existing proteins or change or bind to the DNA and cause the synthesis of different proteins. I know it says new, but really it means different proteins, different new types of proteins that do different things. Okay, so basically the hormone signal comes in and it tells the cell like, now I want you to do this. So you drop everything and do that. Okay, so that's what the signal pathway really is. Okay, so signal molecule binds to memory receptor protein, which triggers intercellular signal molecules, which alter the target proteins, which then creates the response. Okay, so let's take a look at how this works. Okay, first of all, there are two types of signaling. Okay, there's two hormone signaling: lipophilic and lipophobic. Okay, and they do slightly different things. Okay, lipophilic loves fat. If the signal molecule loves fat, that means it can go directly through the cell membrane. Okay, and therefore it will bind to either cytosolic receptors, so receptors that are floating around inside of cytoplasm, or even it will bind to nuclear receptors, the ability to go directly to the membrane and affect nuclear receptors and bind to nuclear receptors, which can then work on the nucleus and change the gene transcription profile. Okay, all right, so I want to set up a little bit of background. Okay, when you have a cell, that cell is under control of the genes in the nucleus. Okay, so certain genes are being made, certain genes are not being made. Okay, this is known as your gene transcription profile. Okay, the profile in which any cell has is their current gene transcription profile. Okay, lipophilic signaling molecules usually alters the gene transcription profile. It tells that it goes through the nucleus, causes changes in the nucleus, binds to certain proteins in the nucleus, which tells the DNA to make different proteins. Okay, this takes a little while, not like seconds. Okay, it takes minutes, hours to change. Okay, but ultimately, the cell changes its gene transcription profile. Okay, for the lipophobic signal molecules, they can't go through the plasma membrane, so they bind to the surface of the cell. The surface of the cell signals, like it has a signal cascade which will go over a little bit. The signal cascade will change the things inside the cell. Okay, but not usually not the nucleus. So the proteins inside the cell do something. They activate something, they activate other things, etc. But they do something. The nucleus, the gene transcription profile isn't really changed all that much. Okay, so that's really the difference between the two. Okay, for the hydrophilic signal, sorry, not hydrophilic, the lipophobic signal molecules, the reaction is almost immediate because all the cellular proteins are already made and they're just waiting to go off, waiting to do their thing. And as soon as the signal comes in, bam, it finishes. Like it does what it's supposed to. Okay. A good example of the lipophilic signal molecules are cell death pathways, which I used to study. So the signal comes in and bam, 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 the cell dies. Like within seconds, the cell dies. Okay. The cell undergoes apoptosis. All the genes that are required for apoptosis has to be already made. Okay. The only thing that's missing is the unlock code. It's like the cheat code. You come in, you turn on the cheat code, bam, everything goes. Okay. All right. So that just being one. Okay. So here, so this is the difference, right? So rapid response, slower response. Okay, because it has to go through the nucleus, make new genes. That usually takes a lot longer. Okay, this one is much faster. Okay, there are four categories of memory receptors: the chemically gated ion channels. That's really easy to understand. The gene protein coupled and receptor enzymes is not. I think we'll skip the integrin receptors. Okay, because they're not that relevant. The first three are. Okay. So here it is. Oh, four. Okay. Okay. So signal transduction. Okay, signal transduction. So the transducer converts the signal from one form into another. Okay, that's by definition. Okay, usually you need two messengers: the first messenger and the second messenger. The first messenger has to go from outside the cell, okay, and it amplifies something inside the cell. So the signal has to travel through the membrane. Once it gets through the membrane, one thing that activates the second messenger. A second messenger, okay, is going to travel throughout the cell. Okay, it is this. So think of the cell being something as big as your house. Okay, receptors are going to be on the outside of your house. Okay, and they're going to be about what size? What relatively size? Like your cell phone. Okay, that signal is going to come through the wall, right? It binds to your cell phone shaped object outside the wall. It's going to come through the wall, but now you have the whole house. So the signal has to go through the whole house. So you need another messenger, not just the cell phone, to go through the whole house and turn everything on. Okay, so to do that, you can't rely on one protein. One protein is small. You have one protein activate 10 proteins. 10 proteins activate 100. 100 activate 1,000. And then you're off the races. Okay, so usually you have cascades of activation steps, and each step is amplified by the step before it. Okay, so one protein turns on 10 proteins turn on 100 proteins turn on 1,000. Okay. Okay. So cell signal. This is the so this is the hormone usually. You have the transducer protein, and then you have turns on a second messenger. The second messenger is amplified. So usually it amplifies the signal a lot, and then it goes down to the protein targets and generate a response. Okay, so you need a lot of like every step along the way, you actually amplify the signal. Okay, you amplify the signal. Okay. So 1 turns into 3, 3 turns into 9, 9 turns into 27, etc. But it's actually more than that. Okay. All right, these are common messengers that you have learned. Okay. All right. Okay. So let's talk to this one. Okay. So signal transduction using ion channels. Okay. So the first thing that happens, okay, you either have an ion binding to the channel itself, okay, and it opens the channel, okay, or you have a signal binding to the G protein, which opens the channel, okay. Either way, the channel opens. When the channel opens, the ions come flooding into the cell, okay, and it came flooding into the cell, then that can cause changes in membrane permeability to sodium, calcium, I don't know what calcium not here, sodium, potassium, chloride, calcium, which causes a change in the electrical signal, which causes a cellular response. Okay, so it's a very quick response, and this is the way your nerves and muscle works. Okay, very quick, very fast, efficient response. However, the problem with this, there's very little modulation. So it's either on or off. There's not a dimmer switch attached to this stuff. Okay, so it's hard to modulate. Okay, but on off, this is the most efficient way. Okay. All right. One of the hardest things to understand is G proteins. Okay, so G protein coupled receptors. Okay, so let's talk about. Okay. Okay, so here is a G protein. Okay, and G protein coupled receptor. Okay, so remember this is so G proteins are usually used by lipophobic hormones. Okay, lipophobic means that it cannot cross the phospholipid bilayer. Okay. Okay, so one single molecule. Okay, that binds right there. Okay, the G protein receptor. Okay, here has to cross the bilayer, and it does. So, and just for your reference, it usually passes 7 times. So 1, 2, 3, 4, 5, 6, 7. 7 will put the other end of it on the inside of the inside of the phospholipid bilayer. So the signal binds here, it causes a change on the other side of the cell membrane, and that will activate the G protein. Okay, the G protein will turn on adenine cyclase. Okay, adenyl cyclase converts ATP into cyclic AMP. Okay, so ATP is kind of linear; it just turns it circular. That circular ATP can now flow through as a second messenger. Okay. Okay. There it is. Okay. Okay. So the hormone is the first messenger. This is the transducer. Transducer, and then the second messenger. Okay, and then the signaling. So the signaling AMP turns on protein kinase A, other things, and that causes other things to respond. Okay. All right. Okay. So take a look. Single molecule activation receptor. Okay, that gene protein can activate adenyl cyclase. Okay, the adenyl cyclase will make cyclic AMP, which will activate protein kinase A, which will cause a response. The other way it does it, okay, the same thing, gene protein receptor binds the gene protein. Okay, the gene protein will activate, instead of activating adenyl cyclase, it will activate PLC, phospholipase C. Okay, phospholipase C will cleave phospholipases, and it'll cleave it into two parts. Okay, one part is called diacylglycerol, okay, and IP3. Okay, IP3 is going to be hydrophilic, so it will float around the cytoplasm and it will turn on other stuff. Okay, DAG will still be attached to the plasma membrane and it will turn on other stuff in the plasma membrane. Okay, so the DAG will turn on other things in the plasma membrane. So it's taking something and cleaving it into two parts. Both parts now have functions. One part that still stays in the membrane will activate the cell membrane to do things. This part that's cleaved off will go into cytoplasm and do things. Namely, it will go into the ER and release calcium. Okay, calcium is the second messenger here, and it will cause other cellular responses to happen. Okay. All right. All right. Hopefully that makes sense. Okay. I'm actually gonna stop right here. I've talked long enough. Okay, so hopefully that makes sense. And then for part two, we'll go through the rest of the chapter.